Okay, we're going to now kind of contrast what are called bottlenecks versus founder effects. They're both mechanisms by which population sizes get reduced. So when we look at bottleneck effects, here's the original population, a bunch of beads. I think you actually do this in lab, okay? And you go through genetic drift and sample, and you get kind of you don't do a bottleneck, but here's the original population. The reason it's called a bottleneck, right, is here's the original population size. It goes through this bottleneck or a reduction in population size, and after the bottleneck event, and then here's the surviving population. Basically what this represents are these are allele copies or gene copies, or they can even be individuals, it doesn't really matter. But basically what you have in a bottleneck is a severe reduction in population size. There are lots of ways that this can occur, obviously, right, through lots of things that reduce population sizes, such as overharvesting, destruction of habitats, destruction of communities, and all these sorts of things. Diseases can lead to severe reductions in population sizes. There are a lot of mechanisms that can lead to bottlenecks. And then you have the surviving population. So the question is, what's the outcome? Well, it's drift. So you get sort of the random stochastic variability and presumably loss of genetic variation. It depends often how long that bottleneck lasts, how severe it is. It also depends on the kinds of organisms you're talking about too. But here's what we can imagine. Here we're starting out with this simulation. Is population of severe bottleneck size indicated by the shaded region. In return, we're starting out with 1,000 individuals in the starting population. This should look similar to graph C, right? At least early on. You still get these stochastic changes in allele frequencies. Populations are becoming different from one another. But here what you're doing around a generation 90 to 105 maybe, what you're doing is you're creating a severe bottleneck, a huge reduction in population size. And so the bottleneck causes divergence between populations. Before the bottleneck, allele frequencies are similar, kind of similar among all populations. What do you notice happens during that bottleneck? What does it look like? It looks like the top graph, right? So it's basically the same thing. We're just kind of looking at, you're imposing, the original graphs had population sizes that were fixed. Here you had a higher population size, and then you reduce it to simulate a bottleneck. And what you get is pretty simple. What happens? Allele frequencies fluctuate much more during the bottleneck, right? If you look in here, if you can follow each one of these lines, there was a dramatic decline in that population. There were dramatic increases in that frequency in that population that went to fixation, right? Depending on where you are, this one went to fixation too. So during that bottleneck, what you had, one population went to fixation A1, the other one went to fixation for the alternate allele. But you still had what happened over here, and what eventually happened over time. Populations still are pretty different from one another. Okay. So after the bottleneck, allele frequencies differ greatly from one population to the next compared to the pre-bottleneck. So the way to think about this is where the extreme genetic drift is occurring, right? You're getting obviously you're getting, and they don't show this, they don't tell you how much the population size is increased after the bottleneck, but it's probably a fairly persistent bottleneck. There's some growth, but it's still experiencing a lot of genetic drift that's occurring here. Okay, let's talk about an example of that. You know what those are? Yeah. So those are elephant seals. They're widely distributed in the Pacific. They range from, there's a northern elephant seal and there's a southern elephant seal. The northern elephant seals had some trouble. And do you know what the trouble was? Especially in the 1800s? Yeah, they were hunted almost to extinction. Okay, so they lived on the beaches of California, a lot of the islands up into northern California, southern California, Mexico, up into northern California. So they lived on beaches, and people liked them, and they got literally almost went extinct. I just read this morning, that in that northern elephant seal population, they went to about 12 individuals, right? They found them off an island in Mexico, and guess what happened? You know, there are museums museums of natural history. I used to work at one that collect animals for various purposes. Guess what they did? They went in and collected all those 12. So anyway, that was in the 1800s, early 1900s. That sort of stuff doesn't go on anymore. But what they did was they went in and harvested those 12 remaining individuals. Fortunately, there was another population that nobody knew about that survived, and there were about 8 to 10 individuals that survived. Huge amount of protection that was afforded to the northern elephant seals after that through conservation efforts, sanctuaries, all these sorts of things, and now they've recovered to over 10,000 individuals. Okay. The southern elephant seals were not over-harvested and provide a nice comparison. So southern elephant seals are primarily along, the Pacific coast of South America. So here, the southern elephant seals are in orange, the northern elephant seals are in blue, and I'm not going to go through all. There are various kinds of measures. Here's what's called the heterozygosity, the fraction of genes showing polymorphism. Does it look like there's a big difference between these two populations? Not from that founding event of about 8 individuals, they're basically homozygous, these southern elephant seals. Here are the number of what are called mitochondrial DNA haplotypes. It's easy to sequence the entire mitochondrial genome right now, and you can determine how many variants or mitochondrial DNA haplotypes they have. What you see again is there's much greater diversity in terms of mitochondrial DNA in the control. This is the control region in the southern elephant seals, and much at all these genes, microsatellites, MHC is a major as a disease involved in disease prevention or reaction. They have much limited the southern elephant seals have very limited genetic diversity, especially compared to the southern elephant seal. This kind of shows there was kind of, I guess, something good about having museum specimens in that they could actually, museums had collected specimens in the 1800s prior to the over-harvesting of the southern elephant seals. And so you could actually go in and get those samples, and in the 1990s and 2000, people went in and sequenced them, okay? So they had what's called a pre-bottleneck individuals, and again, these are the different, again, I'm not worried. What you find here in the pre-bottleneck, there were only 5 individuals that were sequenced, right? Each one of them had 4 out of the 5 had a unique mitochondrial haplotype. So there was a lot of variation in the pre-bottleneck. What happened during the bottleneck and the post-bottleneck? How much diversity do you see? Well, some individuals had one haplotype. Most of the individuals had this one haplotype or this E, okay? So all of these other sort of haplotypes were lost during that bottleneck. Again, all it really illustrates is that severe bottleneck that occurred in around 1890s to 1900 really resulted in a loss of genetic variability. Okay. So those are bottlenecks. Now last thing we'll focus on is we'll focus on what are called founder effects. And this is the case where you have an original source population and you're founding a new population that presumably is uninhabited by the organism you're studying, and so they're founding a new population. And this is what we call founder effect. So again, it's the same thing. What happens is the original population, here are the allele frequencies on the mainland, and a bird carries over 5 seeds that doesn't have this plant on it, right? And it colonizes that, those seeds come along with it, and it colonizes a new island, and it carries 5 seeds, and just by chance, depending on which ones it has, it's very likely you're going to get what's called, with a limited population size. Of course, if it had 10,000 seeds and there are multiple birds with 10,000 seeds, you probably wouldn't see much of a founder effect. But in this case, migrating bird carries seeds to a previously uncolonized island just by chance. These are the genotypes that are associated with those 5 seeds, just randomly picked out of here, right? Well, it shifted from a big R of 0.3 to a big R of 0.6, and that's what we call founder effect. Don't worry about that. This is another very important way that founder effect occurs. Remember I talked about the Pleistocene glaciation? Basically much of the northern hemisphere was covered in glaciers, and that is no longer true. So you have glaciers. Here you have a source population south of the maximum extent of the glaciers, and then obviously as the ice age ended, the planet started to warm, the glaciers retreated, and you obviously have individuals near this leading front that are expanding into this new population. So is this kind of founder effect? This is what's called leading edge expansion. So anytime you have cases where glaciers occur or any sort of disturbance and individuals from a source population move into it, you have this kind of expanding front. And obviously glaciers retreated quite a ways north, right? So it opened up a lot of new habitat, new areas for colonization by various kinds of plants and animals. And so most of Canada, 12,000 years ago, was covered in a sheet of ice. Even most of Michigan, most of Wisconsin, and all those areas were completely covered in ice, and that's true across northern Europe too. Okay. So this is believed to be a very important thing that occurs. Here's an example, boreal spruce forests, right? Here's an example during the wintertime. And you all want to spend a lot of time up there, right? Maybe some of you do. Okay. So here's the current extent of boreal spruce forest in green right here. Obviously, was that here 12 to 17,000 years ago? All of that boreal spruce forest was down in here, okay? And then it's undergone this range expansion from, this was during the Pleistocene, then when the glaciers melted, all this habitat became available in this expansion into these northern regions. So when you think about this, I don't want, your book goes into great detail about this. I'm not too concerned about it, but there are a way that genes move, right? So if you're expanding into Canada, or you're expanding into Sweden or Norway, all these things, or Russia, all this happened all over, right? As these glaciers receded, genes can move and individuals can get there through lots of different mechanisms. And seeds are obviously one of the primary mechanisms by which things move in a particular direction, okay? Pollen is another way that genes move, right? And where does pollen come from? From plants, from males only, right? Whereas seeds have what kinds of genes in them? Well, seeds have, they're the zygote, right? So they have both male and female contribution, right? But in general, what happens, seeds, they don't move as much, do they? Especially compared to pollen. And especially this kind of, how do you think, where does pollen move in these trees? How does it move? Does it need a pollinator, or does it do it a different way? It's actually wind pollinated, okay? So pollen can disperse across long distances. Sometimes in these cases, if it can get into the wind, hundreds of miles. Seeds, you know, they have little wings on them, and they can move, you know, they can expand into that area, but it's the colonization on this leading edge expansion is primarily done through seeds, right? And, you know, these can, you've seen maple seeds, they flutter around, and they can move some distance from the parent, but it's not to a great extent. A lot of other plants use birds and other things to disperse seeds or move seeds, or mammals, and so there are various ways that seeds get dispersed, but in general, seeds kind of disperse locally. So they kind of fall fairly close to the parent tree. Pollen moves a much longer distance. So I don't want to belabor that point too much, but this study really only looked at what's called mitochondrial DNA, and the only way that's dispersed is through seeds. So it's a good way to kind of look at this local effects on genetic drift. So all I really want to focus on here is they went in and determined sort of the mitochondrial diversity in populations down here versus populations up here that they think were really part of the leading edge expansion, okay? And this isn't exactly right because this was actually fully glaciated, but that's okay. So what you'll notice here is where's genetic diversity greater in terms of the mitochondrial genes in this example? Is it this region or up in here? Yeah, So what you can see is here in this population you have 4 mitochondrial haplotypes or variants. Here you have, what, 3, and here you have 4. This has more of the blue haplotype and those sorts of things. So there's some variability among them. What about these populations up here that have colonized through this leading edge expansion? Do they show a lot of variability? Do they show any variability? They show no variability. They're all fixed for the blue haplotype at 100%. What the authors of the textbook state is this is an indication that these have dispersed there and on this leading edge expansion and had a very reduced population size and were founded by what kind of haplotype? The blue haplotype. Okay. Probably if you went in and looked at other genes, nuclear genes, you'd find more variability, but you don't have to worry about that. So what this documents, presumably, is that these populations have very reduced genetic variability because of founder effect as they colonize these once glaciated regions. Okay. This is the last one. Then we'll do a clicker, and then I guess I could entertain questions if you want about the exam, if we have time. We should have time. And maybe I'll go over the key. Should I go over the key to the homework problems? Okay. So what's showing here is all we're really contrasting. This is extremely weak selection. This is a selection coefficient, right? Where it's really, really weak, 0.001. Very weak selection. This is really strong selection, 0.5. Doesn't matter which allele it is. Let's just kind of contrast the green line with the red line. Along here is population size. Okay. What happens even in small populations where selection is really strong? What's the outcome? Yep. So it turns out that what's happening here is, which allele do you think is going to fixation? Is it going to fixation just by chance? It actually turns out it's going to fixation of the favored allele. So what can happen here, selection really works selection isn't very effective in small populations when it's weak. It's pretty effective, right, in small populations when it's strong. But when you go down here you can see the selection is really weak. What's the predominant effect on the fixation of these alleles? It's primarily the size of the population. And you can see even here, even when you have a really weak selection, it's going to take even under population size of 1,000, even low here, you're basically going to get you're going to get a really low probability of fixation of a favored allele. I should make that clear. This is the probability of fixation of a favored allele. Sorry, I should have made that clear. This is the probability of fixation of a favored allele. It works really well when selection is strong, but do you get fixation of a favorable allele when selection is really weak? And population doesn't even matter. Okay, so the key I don't want you to get too bogged down on this, but selection isn't very effective in small populations and when it's weak. It can't really work. So drift kind of overwhelms selection when selection is really weak. Okay, and selection is very effective in even small populations when it's strong. Okay, let's do one last eye clicker. I think this is fairly straightforward. So which of the following is an example of a population bottleneck? A long endangered species recovers and increases in population size. A large population of frogs is greatly reduced due to a drought. Butterfly migrations are disrupted by climate change. A beneficial mutation goes to fixation in a population. So what's the best example, a clear best example of a population bottleneck? This was considered easy by the test makers. Everybody in? Easy, right? What's the answer? B. Yep. Okay. So some people picked A, and a long endangered species recovers and increases in population size. That could be the result of a bottleneck, but it's kind of post-bottle. It's a post-bottle. It's not really an example of a bottleneck effect. Okay. So that's how I would think about that one. Okay. You want those kinds of questions on the exam? Okay. You want me to go through this real quickly? I posted the key, right? So here's the people had, and I knew this would be true. The biggest problem was what's the arrangement of the genes if these two genes are located on the same chromosome in question A. So how do we figure that one out? I think so far I'm almost done. So let me tell you, I thought I was going to try to finish this morning, you know, but I got up at 3 in the morning for you all to try to accomplish it and get it done, you know. But I just couldn't get them all done. So I got about 10 more to do. What I will do is I will put them in the front office in sciences with Charlotte Ann. So if you want to come by at any time to pick them up, you can. Don't go in and say, Charlotte Ann, oh, I want to pick up 10 from every student. She can't really do that. So we can only give them out to you individually. It's a privacy issue, okay? So if you want to pick them up, I'll send an email, Sciences 1100. I should have them done and entered in the Moodle gradebook sometime, I hope, this afternoon. So come by tomorrow. I'll bring them to the review sessions if you want to pick them up. Okay. So if you don't want to stick around now, you can leave. So let's kind of go through. So there's the answer. How did I come up with big A and small b being on the same chromosome and small a and big B located on the other chromosome? We did come up, we rejected the hypothesis of independent assortment. These genes were linked, observed were not even close to expected. What's the first thing you need to look at? Which are the most frequent genotypes, right? When you do this kind of test cross, those are what we call the non-recombinants. The recombinants are the rarer ones. So you kind of have to just make that leap and understand that the non-recombinants are always going to be the most 279 and 303. These were less in frequency. Okay. How did I then get A and B, big A, small b, small b from this? So just kind of look at it this way. One of the parents contributes only the recessive, the small a and the small b, right? You'll see in each one of these genotypes, here's the small a, small b, here's the small a, here's the small b, here's the small a, small b, small a, small b. All you really basically do, and you can, that's in the non-recombinants, that's the parental combination. Is big A, small b, and small a, big B. How do you get these two other genotypes? Through crossing over. Where does the crossing over event, where's the crossing over event have to occur during meiosis? Over here or in here? Yeah. So the crossing over event has to occur in here, and then what are you going to produce from that? What kind of gamete? So all of these individuals are going to produce big A and small b gametes and big A and big B gametes, right? Those are the only two kinds of gametes that parent's going to produce. if you do a crossing over event, what are you going to get? Big A and big B being a new gamete, and small a and small b, there's the big A, the big B, small a, small b. Those are the recombinants. This was the really hard one. And I think I didn't take off, if you didn't get this, I didn't take off any points. Do you think these genes are tightly linked? What you have to do is you kind of have to figure, I was just seeing if anybody, they actually aren't. Because you actually get a fair number of recombinants. So the idea here is that these two genes are probably separated by quite a bit, so that crossing over, if it just occurs at random, is going to occur with a wide space between them. If these things were like right next to one another, you have a very limited range over where the crossing over event can occur to produce a recombinant. And that's where you get really small numbers of recombinants. And I kind of remember I talked about that in class where I had one individual. I had two recombinants total. That would be the case where those two genes were really tightly linked on the same chromosome, really close to one another, and you can rarely get a crossing over event. Okay. So I said genes are located on the same chromosomes. Here's how they're arranged. They're not tightly linked because there are many recombinants. That's a pretty high, and I didn't take off any points there. This is about a 20% recombination rate, which is really unheard of. Everybody did pretty well on this one. A lot of it, I found a lot of times you just made an error. You said instead of putting 0.564, you put 0.364, so I only took off about 5 points for that. No, I don't take off any points for like errors that clearly you're just kind of in a hurry, or I do this all the time, so how can I hold that against you? So if you did the calculations correctly, you came up with the wrong number, I don't really care. As long as you know the process and how to do it, that's the most important thing. Okay. But I'd say as you go through the exam, a lot of times check your work. So some of you didn't do it and you didn't have allele frequencies summing up to 1 or genotype frequencies summing up to 1. Always use that as a check as you do the calculations to help you to make sure you're doing it probably correctly. Okay. So make sure you can definitely do this chi-square test. So here's the key. The hardest part was, again, what was the hardest part for you? If there is a significant deviation from Hardy-Weinberg, what could explain this? And again, now you're armed with drift, inbreeding, all kinds of selection. Hopefully you'll be able to better kind of interpret these things. But here are the key things to notice. Here are the observed numbers of each, right? 250, 800, and 200. Here are the expected under Hardy-Weinberg. Obviously, you rejected that it's in Hardy-Weinberg because, these aren't even close. Okay? High value chi-squared, degrees of freedom 2, p is less than 0.01. You firmly reject that the locus is in Hardy-Weinberg equilibrium. What you have to do is you got to kind of compare the observed and expected. And what I was hoping you would find, and some of you did, and this was the hard part, you observed 800 heterozygotes. What were the expected under Hardy-Weinberg? 624. So you observe more heterozygotes than you expect. What about the 2 homozygotes? What do you see there? You observe 250, but you expect 338. So there's a deficiency of homozygotes. And again, even this one, you observed 200, but you expected 288. What kind of selection could explain that? And if you all just said mutation, random, non-random mating, selection, I gave you credit at this point. But probably on the exam, I might look for a little more insight. Yeah. Yep. So the most obvious explanation for this would be you find more observed heterozygotes than expected because they have a selective advantage, heterozygote advantage or overdominance. That's what I was really kind of looking for. Okay? Does that make sense? And then lastly, people did pretty well on this one. It's kind of, I won't tell you the really quick way to do this. Every semester I find somebody says to me, why are you doing this long convoluted thing when I can do it this way? But anyway, I shouldn't have said that. Okay, everybody did fine on this. The key thing here, this was a form of dominant selection, right? big D, and the heterozygote had a fitness of 1. That's dominant selection. If the heterozygote had a slightly reduced fitness compared to the homozygous dominant, but high above the small d, would big D go to fixation faster or slower? Big D. It'd go to fixation faster. It would increase each generation. If it's co-dominant, that big D is going to go to fixation faster, and the small d is going to be eliminated faster. Okay, so as you think about that, right, and you know, I'll be doing a problem similar to this, but who knows, it might be co-dominant. So, and what I would suggest you do is do this exact same thing with a partner or somebody in a group, right, and change the fitnesses to co-dominant. So reduce the fitness of the heterozygote. If it's 1 and then it was 1, make it 0.8, and the other homozygote keep the same. Then calculate. And what you should see is big D increases more under codominance than dominance at these frequencies. Yeah. Yep, same thing. Yeah, there are about 2 other terms that we use interchangeably. Yeah, so it's always the case where the heterozygote has intermediate fitness. Yeah. Okay. Does that make sense? Or okay. So really, you know, those are going to be the kinds of work problems on the exam. Okay.